All NEET aspirants, you are listening to podcast Botany for NEET by S. Ramakrishna in this continuation of the topic that is biotechnology principles and process. The use of live organisms, enzymes from the organisms to produce the products useful to the humans, what they call biotechnology. A home kitchen is a mini biotechnological lab. A housewife certainly considered to be a traditional biotechnologist who unintentionally uses the microbes present in the curd as a starting inoculum, converting the milk into curd, that is raw material milk into curd. Traditional biotechnologist. Curd making, bread making, wine making, all microbe mediated process is a biotechnology. Now, biotechnology used in restricted sense today, that is, the production of curd, bread, and wine in large scale at industrial levels by using genetically modified organisms. What they call it is biotechnology. So, previously we used microbes without any intention, but later genetically modified microbes with intention. Next. Biotechnology includes, that is, in vitro fertilization, test tube baby, using the vaccines, the DNA based vaccines, synthesizing the gene and using of the gene, and defective gene is replaced, that is the correction of defective gene by gene replacement therapy. All it is included in case of biotechnology. Next. EFB, European Federation of Biotechnology, or EFBT, encompass, that is combines traditional view as well as modern molecular biotechnology and gave the definition for biotechnology. Integration of natural science, physics, chemistry, biology, organism, cells, molecular analogs, likewise molecules like DNA, RNA, to produce the products and use it in the process, what we call biotechnology. If I elaborate the definition, exploitation of microbes, that is utilization of microbes, their properties, their parts at industrial level to generate useful product for human being, what we call biotechnology. Next, biotechnology based on number of branches, but two core techniques which is the basis for birth of modern biotechnology, that is genetic engineering, bioprocess engineering. Genetic engineering, manipulation of the gene, tailoring of the gene as we decide, that is alteration in the chemistry of DNA or RNA, what we call it is genetic engineering. So we change the genotype. If you introduce it into organism, change in phenotype, what we call genetic engineering. Next, bioprocess engineering, otherwise tissue culture. Ambience for sterility is created through chemically engineered process. So ambience not for look, ambience for sterility. That means it should free of other microbes to enable the growth of particular microbe, whether prokaryotic cell or eukaryotic cell. It should made to grow in such an environment, sterile environment, free of other microbes to produce large scale products, that is enzymes, vaccines, proteins, like that. So ambience for sterility is created through chemically engineered process in this step, bioprocess engineering. Next, conceptual development and the principles of genetic engineering. What is the actual principle that lies in the development of a branch, that is genetic engineering? If we take meiosis, the crossing over, exchange occur between non-sister chromatids of homologous chromosome in packaging, what we call crossing over, leads to recombining of the genes, leads to variation. Here, sexual reproduction having advantages over asexual reproduction, because in sexual reproduction it promotes variation, It permits variation. That means new formulation of genetic setup leads to variation, which may be useful to organism, or it is some of may be useful to organism or population. And asexual reproduction permits genetic integrity, no variation in case of asexual reproduction. In traditional hybridization in plants and animal, inclusion and multiplication of desirable as well as undesirable genes occur. And this extravagancy, this limitation, it can be overcome by RNA technology, gene transfer, gene cloning, that we can transfer only desirable gene into the target organism is possible without inclusion of undesirable gene. It made it possible through genetic engineering. Next, we can transfer a piece of DNA into alien organism. It would not multiply. What might be the reason? Whatever the segment of DNA transferred, that might lack origin of replication. Second, it might be introduced into cytoplasm, not the integral part of the chromosome. So there is no multiplication of this foreign DNA in alien organism. If this piece of the DNA integrate into the host chromosome, it multiply along with the host chromosome, because it becomes the part of the chromosome in case of the host. It replicate along with its DNA because of what it is, chromosome having DNA, DNA having ori. So any DNA linked to ori can multiply and produce indefinite number of copies of linked DNA or gene. What do you call it is gene cloning, or getting templates of the DNA. Next, so any piece of DNA, it should become the part of the chromosome. The chromosome having DNA, it must have ori. It is not inserted into the ori, it is linked to ori. If it is inserted into the ori, means ori might be deleted. So it is linked to ori. So it is capable of producing indefinite number of copies controlled by the ori of what it is, the particular DNA. Next, construction of first recombinant vector by Herbert Boyer and Stanley Cohen. Because altered plasmid introduced into the host cell by Herbert Boyer and Stanley Cohen. The gene encoding for antibiotic resistance was cut out from the plasmid of Salmonella typhimurium by using restriction endonucleases, which are called molecular scissors or molecular scalpels, which cleave the plasmid DNA. That is, gene encoding for antibiotic resistance was cut out. That is phosphodiester bond. It breaks the phosphodiester bond. So single-stranded sticky end formed at the end of double strand by using restriction endonucleases or restriction enzyme. Next, so as to express this gene encoding for antibiotic resistance in host cell, first it needs to be transferred to the host cell. For this transfer, it needs a molecule, a vector, a carrier. The vector or a carrier is a DNA molecule which carries the gene encoding for antibiotic resistance to the host cell for multiplication. What do you call it is gene cloning. That is cloning of antibiotic resistant gene. So the plasmid or vector selected from the native plasmid of the strain of Salmonella typhimurium, whatever the enzyme used to cut the gene encoding for antibiotic resistant, the same enzyme used to cut the vector DNA. Sticky ends are formed. It is a linear. So first linear fragment having single-stranded sticky end, gene encoding for antibiotic resistant, and the vector DNA, which is ligated from one end to other end-to-end ligation by using DNA ligase, what we call molecular glue, as it links phosphodiester bond, and this linear DNA again becomes circular double-stranded DNA. Now this plasmid DNA, it is with the insert, that is gene encoding for antibody resistant. So it is normal plasmid. It got inserted with the foreign DNA gene encoding for antibody resistant. Now this vector, what do you call it is? Recombinant vector or chimeric DNA. That is vector with the gene encoding for antibiotic resistant. This recombinant vector transferred to host cell, that is E. coli, similar to Salmonella. How you can say similar to Salmonella? E. coli is a prokaryote, Salmonella is a prokaryote, but it differs. Salmonella is a pathogen parasitic. E. coli, it is symbiotic. In order to express the gene encoding for antibiotic, it should be transferred to E. coli, Escherichia coli. So gene encoding for antibiotic resistant replicate independently in case of E. coli and produce indefinite number of copies of gene encoding for antibiotic resistant. What do you call it is? Cloning of antibiotic resistant gene in E. coli. So it replicate independently in case of the E. coli, but dependent on metabolic machinery, DNA nucleotide as well as enzymes, but replicate independently of the coding strand or the main genome. So what do you call? Cloning of antibiotic resistant genes in E. coli. Again 3 points. The first recombinant DNA achieved by Herbert Boyer, Stanley Cohen. Unaltered plasmid transferred into the host cell by Cohen. Altered plasmid transferred into the bacterial cells by Herbert Boyer, Stanley Cohen. Which enzyme used to cleave the DNA to get the gene encoding for antibody resistant restriction endonucleases? Restriction endonuclease breaks which bond? Hydrogen bond or phosphodiester bond? Phosphodiester bond. Whether selected main genome or plasmid. The vector is nothing but plasmid. Expressing system is E. coli. So gene encoding for antibody resistant from Salmonella. Vector it is native plasmid of Salmonella. Expressing system it is E. coli. Next, in order to genetically modify an organism, there are 3 basic steps. First, we must identify DNA with the desired gene. Next, it should transfer to the host cell. And transferring and its maintenance is important in case of host cell. We should ensure the condition to maintain in case of the host cell. And transfer it subsequently to the progenesis, that is important. So the 3 basic steps to genetically modify an organism. I am signing off from this episode. In next episode, we will see the tools of recombinant DNA technology. So listen with concentration, connect to the concept. If you feel the strategy is right, subscribe and share to the person, those who are seriously preparing for NEET.